So this is Microloop. Microloop is the app that I developed for this 16S data analysis, okay? Then once you got your OTU table or ASV table and your metadata and your taxonomy table and everything ready, then you can bring it to the count data here. You don't have test data with yourself, but instead I have already built in some test data that you can load here. Just click on this button, load test data, and then something should appear here. Don't overwhelm my CPUs, but if you download, click on this download data.zip, and then let's go up here and look into the shape of this data. That's it basically, and you also should get something like this. And then when you have your data, your own data, okay? Your own data should exactly look like this. At the top here, this shouldn't be empty. It needs to have a name. Let's call it ID. Anything. And what is the importance of that? If you unload your own data, the column, enter the column name to be used as the row name. That's the name of the OTU's column, which we call it ID.
So the first column.
The first column that the software is going to use as your row name. Now you need to know to use it here. Okay. So I'm going to just click and open this. You don't do anything. Okay, just for me. I want to see if something happens. Actually. Again, you need to have a name for that first column or that column that corresponds to the column name of your OTU table. And here is this one, which begins with barcode 01 to 36. And I just call it ID again. Doesn't matter. Call it barcode, whatever. Call it your name. Right. And then here, these are all the information that I have about my study. Okay. For example, in my treatment, I have one group which is called naive or control group. Another treatment, we have given them protozoa. This was a study is a case for inflammatory bowel disease. In the weeks, we also have week 0. In it, the treatment is confounded, is nested. Okay, we need to know about the design of the experiment, which obviously some variants might come from the week itself, which later on I will show you how to factor it out. Okay? Or put around the random error, something that you can reduce. There is a tick here, there is a checkbox. Data has a header. Our data has a header, which usually should have a header, and yes. If it doesn't have a header, you should set it as no, okay? Mm-hmm. Metadata, again, you load it, and then if it's CSV, that's the one. And CSV files, TXT, no problem, but if it's CSV file, you need to know your delimiter. Is it comma, is semicolon, whatever. And then you just choose it, and then give the name of that column that you want to use as the row name, sample name, in this case ID, whatever. And then data has a header, yes. Taxonomy table. There are two types of taxonomy data. One of them is parsed, one of them is non-parsed. Ours is parsed. And what it means is to be parsed. This is a parsed, clean taxonomy table. Okay? Again, I give it a name here. ID or OT or whatever, okay? Completely arbitrary. But this is parsed. Let me show you an example of an unparsed data. How messy. How messy. But if you look at it, it's only two columns. This column, and this column, right? Inside this column, you see that we have all concatenated taxonomy levels. K for kingdom, bacteria, semicolon. P for phylum, semicolon, Clostridia, all the way down to the species level. Okay? This is unparsed, and that's important. When you have unparsed data, you should not have a header, okay? We don't have a header. You only have two columns. This column and this column. If you want to parse your data yourself, you could do it, whatever, but why would you do it when the app can do it? So usually our output is unparsed. It needs to be only two columns. One column your unparsed taxonomy name, and the other one is OTU table. Sometimes it has a header, sometimes it doesn't have a header. So what we do here, we need to specify here. First and foremost, you upload your taxonomy data. Data has a header. Mine doesn't have. Maybe yours have. If yes, you need to know what column you should use for OutU tables, the OutU names, the first row, okay? And that's what you put it. My data doesn't have a header, and by default, if it doesn't have a header, you just put x1, okay? Unless your data has a header, and you know which column. If it doesn't have a header, 2 column, unparsed, just give it x1, okay? And here, is your data parsed? My data is not. Yes or no, okay? No. Upload phylogenetic tree, if you have, not necessary, but it's good for phylogeny-based analysis. And then upload representative sequences, not necessary at all, just for demonstration purposes. But anyways, now we don't do that. You know how to load your own data. If you have filled this form, okay, this form is necessary to have, requires you to have one OutU table, one metadata, one taxonomy table. Necessary, okay? The rest is optional. And then again, you collect data, okay? Load test data is only for this demonstration purposes. Your own real world data is collect data. That's it. Now let's take a look at this menu bar, sub-menu bar. We have 1, 2, 3, 4, 5, 6, 7, 8 sub-modules in this count table module. The first one is just input. Basically, you could also subsample it, but we have good CPUs and good resources, so we don't do that. Let's continue with the full analysis. This is an overview of your metadata. You can sort them. You can search for whatever. You have an overview of your count table. And a basic plot of the distribution of your counts, basically. And you can see how tailed this data is. That's what I was talking about. It has a lot of zeros. There are a lot of taxa that haven't happened in many samples, and some of them happen. Okay? So, first of all, we don't have negative data. This is discrete distribution, okay? So we have a lot of zeros. This is very sparse, okay? 0 or some positive values. If you hover over these bars, you can see the count, the number of the zeros. You have 22,205 incidences of zeros across the whole data, okay? And then it goes all the way down. And you also have something here, some little counts, okay? I want to say something, and I don't want to touch upon again. You can download these plots at any format and any quality and any dimensions. If you want to have it as interactive as this is, you just click HTML, open HTML, and then you have the responsive plot like this for demonstration purposes and presentations. Okay? You can also download it as PDF, JPEG, or PNG. You can also decide on the dimensions, whichever dimensions, whichever quality you can desire for your publication. This one is also good for having an overview of distribution of the reads. This is what we call sequencing effort or sequencing depth, okay? This is it. So it's telling you that this barcode, you can also hover a little bit here, play with this bar and see how many samples fall within your criteria. And if you want to reset this plotly plot, just double click on it, okay? Do you have the app? So it tells you how many low, how many actually good you have. It's good just to have some understanding of it, okay? Maybe it's also good to have later on if you want to do rarefaction. I'll tell you what that is, for those people who know. This might give you an idea to select a threshold for it. Right? Again, we can download and not tell about it anymore. Anyways, now that we are ready, we see how our data looks. Now we actually start our pre-processing, which is very hefty and tedious and a little bit risky job, risky business. The first thing that we do here is we do a raw cleaning. This is a 16S data, meaning that it's only for bacteria. So I select those who don't belong here. This is misclassification, misassignment. This is error. Eukaryote, unassigned, remove it. Okay? Keep bacteria by deselecting. That's how mine works. This is also another algorithm for checking whether your different taxonomy level, at different taxonomy levels, whether you have some sequences that happen only once, and if so, you can remove them. Okay? Again, this decision is, you need to know what you're doing actually. So we set it to file level. You can also see if there's anything here suspicious true, then you can select this to remove or no. Mm-hmm. So we don't have any suspicious, but if you go, for example, to family level, let's see what happens. In family level, you have one, but this is already, I don't think that's an erroneous. You see this is a mice data, and then you can see if Mycoplasma metacia does exist in mice colon. Yes, it does. I checked this before. So I'm not going to remove it, okay? This is how you need to look at this. Sometimes it really doesn't exist. Sometimes it comes from, I don't know, some lake or something, or it's strict aerobic bacteria. Maybe it should not belong to a healthy gut of the mice. We don't know. You need to check this and know what you're doing here, okay? Mm-hmm. Anyways, I'm going to set to no here. I don't want to remove that. And then here it goes down to the to this criterion. So I'm saying how many samples one taxa should happen so that to be considered an actual biological variant. But anyways, like I talked about, if you have a study design, like control study design with treatments and everything, you need to be a little bit lenient of selecting yes to this one, because that's okay. But if you have a pilot study or you have collected sample from some harsh environments, then don't set this one to yes, okay? So here I'm saying at least one sample should have this taxa for it not to be considered an error, okay? And then with this criterion, it found 5 of them. If you, let's say, increase this, it might go for really a lot. So if I increase this criterion, it found 40. I don't want to do that because I don't have that many samples per each group, okay? So I just stick to the default, 1, and then select yes, I want to remove it. Down here is about singletons, and this is abundance-based singleton finder, which, where is that file? Yeah, if you go here, we say we have 2 situations. If a taxa proportionally happens 100% times in across all samples, this will give us a relative abundance of 1, okay? Just keep zooming. You can find this bugger here. So you have 6 of them, which is relative abundance of 1. It can tell you 2 things. One, either it has occurred across all the samples, or it has occurred only across one sample. And in this scenario, the algorithm checks for the second. If the second holds correct, then we'll remove it. Otherwise, it doesn't show it as a singleton here, okay? So no worries. Here you can be, again, very careful, depending on your design. I just selected yes, and then either you can apply or skip processing. Apply processing. If it's a pilot study, you can skip, you can set most of these to no. Then it's telling you that from out of 697 taxa, you have 666 taxa. Okay, so we have removed some of them. And this is an overview, which is an output, standard output of phylostick object. It's telling you inside OTU table where all your taxa have been curated to 666 number. You have 36 samples, and in your samples you have 10 variables, okay, in your sample data. And you have representative sequences, and also you have a tree with 666 tips and internal nodes, which is the last node is the root. And then, again, I want to mention one time and no more. You can also download those data that you have curated or have done anything on it. So in each step that you modify the data, you have the possibility to download it if you want to do more thing about them, okay? Okay, now that you have pre-processed, you can see that there are these options will appear. So eventually when you go here, it's empty, but bar plot you can see already has appeared. Differential binary analysis has already appeared. Association analysis has already appeared. Phylogeny based analysis has already appeared. We have done pre-processing, okay? Then we go back to alpha diversity, and first and foremost, we need to take into consideration that when you sequence samples, let's say you have two absolutely identical samples, the femtomal amount of DNA the same sample, the same barcode, everything is supposedly to be the same. And if I sequence it, in an ideal world, we need to get the same results. Mm-hmm. But it doesn't hold in the real world, okay? We have pipetting errors, we have PCR errors. If your library preparation requires PCR, you have many biases. And most importantly is the sequencing machine. So in Oxford nanopore technology, when you have a plate with a lot of pores, like about 3K pores, whatever, not every pore has the same quality, okay? So two samples go through two different pores, they are going to be a little bit different. So this within sample variance, we need to count for somehow, okay? So let's say two samples have 10 copy number of 16S gene, and two samples should have 10 copy number, but one of them shows 5 copy number, the other one shows 6 copy numbers, or 7 copy numbers, or actually 10 copy numbers. What we do in this case, we know these samples are identical and they need to be identical. We could do random sampling up to a certain level. So this is alpha diversity. So we calculate alpha diversity for each depth, for each rarefaction depth, and then we see, okay, if I do this, if I rarefy my reads here, what happens? Okay, so what happens, rarefaction is a trade-off between keeping the number of taxa, which is your depth, and the number of samples, which is this line. So if we zoom in a little bit here, zoom a little bit more, and if you hover on this, it will show you which sample this one is, for example. But it should, yeah. Now something appears. But you can see here, let me go a little bit here. I think this is more interesting. This is very interesting. So you can see if I choose to keep 5,000 taxa for all the samples, you have it now? Perfect. What happens here, I'm losing this sample. Because this sample apparently doesn't have 5,000 read in it. But I keep the majority of my samples. I'm also losing this sample. You need to look. Again, rarefaction is not recommended for pilot studies or for samples coming from rare conditions, okay? Don't do this. So instead, now let's say that we chose rarefaction and hit apply and look at the distribution of before-after rarefaction, okay? Rarefying, it doesn't actually influence the distribution of your data as such, okay? The skewness, the tail of your data. So when I click on apply already, you can see that before make it doesn't really influence much. Of course, you are losing some, and if they are under zero count, they will make the data look a little bit more good. But you can see the data is still heavily tailed. And if you zoom on each of those bars, you see they have been, of course, grouped more because the bar is getting thicker, but still under zero count. So for this reason, I don't think this is a very wise choice to select. Instead, we all go and collect on cumulative sum of scaling. And if you want to combine it, say, okay, I want to bring all the samples to the same depth and do the cumulative sum scaling, that's probably the best, okay? Mm-hmm. Another one is central log ratio transformation, and the combination of this with rarefaction. For this demonstration, we just select CSS. This is something we want to bring this a little bit down, make it a bit not like a uniform distribution, but ideally, right? I'm not talking about normal distribution, I'm talking about uniform distribution here. Again, don't want to mention it, but you can download this. But now that you have basically did some normalization or standardization on your data and the depth, now you can visualize this alpha diversity indices. So alpha diversity is when you are looking into the mean or variance, of your data within the groups, okay? They have observed, which is just how many texts are there, how many. We call it richness. These two are for richness, chow1 and this. Chow1 takes into account singletons and doubletons as well. This one is basically the pure raw number of texts that you have. Then you have something for diversity-based index, which is like how evenly, for example, these texts are distributed. This is very uneven because you have, this one actually is rather even, but this one is very uneven. You have a lot of rare texts out. A lot of them are highly expressed, some of them are medium expressed. That's not evenly distributed. So the Shannon entropy of it will be low, okay? So the higher the Shannon, the more even that your texts are within your samples, okay? Now calculate this alpha diversity, and then you look and see, okay, now that I have the alpha diversity for each sampling group, how do they look compared this group to the other group? First and foremost, I need to also add something that we have the phylogenetic tree here. So you can also bring into account the phylogeny, phylogenetic based diversity, which is faith phylogeny diversity. So click on faith, and let's just visualize our data. See how they look, okay? We automatically recognizes the header of your metadata. And then here, let's just visualize it on the treatment, because treatment is of my interest, okay? Everybody's there? No. Great. Now you can see that you have three facets, and these three facets, you have two information. One, the mean of the naive, and for protozoa. Okay, this is, take it as a control group, and this is the challenge group. And then you have this density, they call it violet blood. It looks like everything but violet, but it's a density. It's good for showing the density of your data. Okay, the sample, how densely, how evenly they are distributed for each group. And then actually, if you click here, you can also make these densities disappear and just take a look at the median. Yeah. One more thing that is important. I know I have done the analysis of this data, and I know that weak has some sort of variance that I'm not interested in. Okay, I'm interested in only treatment. So what I do, I also look to see if the weak, if weak has an effect. So if you choose weak here, you need to only go for one of these things. Okay, but if we want to have it multigraded with 4 variables being informed, you can do like this: type variable, tilde sign, and weeks. And then look what happens. It automatically, it's all weeks, right? So, maybe, yeah, there we go. So this is very, very cool and a very informative way of visualizing your data. So what we have here on the rows, so it's a multifaceted grid that you have information about your alpha diversity, information about weak, okay, and information about treatment. Isn't it cool? So this is almost like visualizing a matrix model. Now I'm going to base on this. If you deactivate this densities, just take a look at the box plot, the empty box plot. You can see that in week 0, we already have a bit of a difference in the protozoa compared to naive. And then in week 1, we have almost the same, just by naively looking at it, okay? And in week 2, which is like the 3rd week of our experiment, the protozoa, the mean of protozoa for almost all of these alpha diversity indices is actually lower than the one for naive, okay? So is it treatment or it's weak effect? I hope it's treatment. What we do here, we basically set up our generalized linear mixed effect model and take the variance that comes from weak and put it on the random effect, okay? So in this case, we go to the GLN M, generalized linear mixed effect model for alpha diversity. You can use the typical LMER for Gaussian distribution. Let's just do it with this, and then we can also try it with GLMER. So this is the syntax of R. We say we are interested in the fixed effect of treatment plus the random effect of the random intercept of weak. Holy Christ, I think we are running out of time. Let me speed up. So in this case, this one will be ignored. The rest of this will be ignored. You just click fit model and see what happens down here. Give it time. Okay, great. Now you have your model extract. There are two things that you need to see first and foremost before looking at this, but then you can go down here to see the fitness of this model, okay? To see how good this model is for this data. So basically it gives you some post-hoc statistical analysis on the observed and the fitted data and the residuals and the observed and expected with the deviation from the residuals. So none of this is significant. It also gives you a test for deviation within group. If you don't have any red marks, nothing significant, you can say that, okay, this is actually a good model. But now, what if I want to see how my GLMER works? If you want to have GLMER, you need to have a gamma family link. It could be identity, which doesn't do any transformation of the response, or it could log transform your response. Let's stick to the log and fit the model again and see how it looks. See, we have, with this model, you have a little bit of a deviance basically within the groups, which makes the variance of your residuals unhomogeneous. So you can stick to the LMER and statistical outputs for this. There are two things to know. This is the model summary with mean differences. Okay, we are comparing the means, not variance, in this example. This is for CHA-1. Okay, the alpha diversity for richness, and then you have here the intercept for your linear model, and then the slope for your treatment. Okay, so if you don't give them anything, this should show. This is protozoa. Accept it from me. Okay, this is protozoa compared to the naive. If you don't give them anything, okay, this is the base mean. You have 498 taxa. Okay, but if you give them protozoa, already you have an increment of 16.8. Okay, 16.8 is your beta 1 in the linear coefficient. This is the estimate, and then you have a standard error, the deviation of your estimate by standard error. It gives you the t statistics, and then it gives you the p-value based on the t statistic distribution, and also this gives you a confidence interval in it, which contains 0, meaning that you basically cannot accept this difference. So the difference is also not statistically significant. Thank you. For CHA-1, for me, Shannon, the same scenario. That's it. But again, remember, the estimate is not 0, and it's important. Okay? So protozoa is doing something, is increasing the diversity and the richness of the bacteria. This is mean. Oh, sorry, faith. We also have faith, phylogeny based. Again, the same scenario. Okay? Now we look into the variance. So this was the analysis of mean, and now we want to see the differences of variance. And this is based on a chi-square distribution with certain level of degrees of freedom and chi-square test. Now we could go to beta diversity. We have looked into diversity within samples. Now we are going to see the groups, okay? The group between groups. I'm not going to talk more about this transformation of data, but you have different ways of doing it. Let's just select log transformation. We have central log ratio, robust central log ratio. We have variance stabilizing transformation, and so on and so forth. But most and foremost, I forgot also to tell before, you need to remember what type of data do we have. It's relative, right? Because we don't know the actual copy number of 16S. So if this is a bacterial cell, and if this is their chromosome, some of the bacteria have several copy numbers of 16S, some of them have only one. But the data, what we see, we assign each sequence to one bacteria, right? So we don't have information about the copy numbers. So if we have 10 Escherichia coli, for example, according to our sequencing results, Escherichia coli, let's say, has 5 copy numbers. If we stick to our results of sequencing machine, we mistakenly overestimate the number of Escherichia coli. But Escherichia coli only has 5 copy numbers. So if you divide 10 by 5, in reality, you have only 2 cells of Escherichia coli. That's why we have a proportion of copy numbers of 16S. We call this data simplex data. It's proportional data. It's relative abundance, okay? For ecological studies, it's fine. If you have corrected for this copy number, you can choose absolute. That's it. For vector diversity, if we go back again, there are 2 ways. It says, do you want to take the previous steps, normalization and standardization, which I do, or you want to just take the data as raw? You could also do that. Nothing wrong about it, as long as you know you do some transformations here. Anyways, you want to agglomerate your taxa here, okay? Agglomerate it, because in species level, if you look at your OutU, you have a lot of, let's say, Salmonella, whatever, or Salmonella, a lot of copies. So what do? We agglomerate them. We say take all of them who have the same name, give them one name, but give me the number of them. So we have 3 Salmonella. That's called agglomeration. We do agglomerate here, okay? You remember. You want to agglomerate here? I said yes. Which level? Always species, okay? And then you go down, you do the sum visualization. Sorry, I need to apply first. This is beautiful. This is so powerful. You see, this is what I mean. Uniform distribution. But don't tell anyone that you heard it from me. But this is good to have it this uniform distribution of data from this screw till that. Okay? So it brings up the ones who are less expressed, brings down the ones who are highly expressed.